Viral hepatitis causes inflammation of the liver, leading to both acute and chronic diseases. If left undiagnosed early or without treatment monitoring, these viruses can cause life-threatening conditions like liver cirrhosis and liver cancer. Among the viruses that cause hepatitis, Hepatitis B and Hepatitis C are known to be the most common and fatal. According to CDC data, there are approximately 250 million HBV carriers or chronopatients and about 50 million HCV carriers or chronopatients worldwide. Additionally, about 3 million people are newly infected with HBV and HCV each year, making viral hepatitis one of the well-known diseases that threaten human health. To determine the treatment and monitor its effectiveness for hepatitis B and C, it is crucial to perform a quantitative test to measure the viral load in the body. Quantitative viral load testing plays a very important role in various aspects of patient management, including evaluating disease activity, deciding the timing and effectiveness of treatment, and assessing infectivity. It is especially necessary to accurately quantify even a small amount of virus. Bioneer has developed and offers the AccuPower HBV Quantitative PCR Kit for HBV quantitative testing and the AccuPower HCV Quantitative PCR Kit for HCV quantitative testing. The HBV and HCV quantitative test kits are in vitro diagnostic medical devices that quantitatively analyze hepatitis B and C viruses in plasma and serum samples. These kits incorporate Bioneer's unique dual hot start technology, enabling the quantitative analysis of even minute amounts of virus. For user convenience, Bioneer's HBV and HCV quantitative kits are provided in a premix format. These kits are used with Bioneer's molecular diagnostics system, ExiStation. ExiStation consists of the ExiPrep system for automated nucleic acid extraction using the AccuLoader and the Exicycler for real-time PCR. AccuLoader provides a guide for dispensing samples at designated positions. The automatic setting of dispensing positions prevents user errors during sample dispensing, and the anti-contamination shield and filter prevent cross-contamination. ExiPrep 16DX and ExiPrep 48DX are automated instruments for nucleic acid extraction from samples. The ExiPrep 16DX can extract nucleic acids from 16 samples at once, while the ExiPrep 48DX can handle 48 samples, and both automatically dispense the extracted nucleic acids into the real-time PCR kits. Both instruments are equipped with a UV lamp and an anti-contamination shield to prevent errors in the results. The ExiPrep 16DX can be connected up to 3 units, allowing for flexible use depending on the scale of the laboratory. The ExiPrep 48DX has the advantage of automatically punching the nucleic acid extraction cartridge and processing a larger number of samples at once. The extracted nucleic acids are automatically dispensed into the HBV and HCV quantitative test kits, and the tests are performed on the real-time PCR instrument Exicycler, which then provides the results. The clinical performance of the HBV and HCV quantitative test kits used with the ExiStation was evaluated, and the results showed a quantitative correlation of 2 greater than or equal to 0.95 for both. Bioneer's HBV and HCV kits have received approval from the Korean Ministry of Food and Drug Safety, are the first in Asia to obtain CVDD List A certification, and are listed on the Global Fund procurement list. Bioneer's ExiStation for HBV and HCV diagnostics provides a solution for the quantitative testing of viruses in samples from patients infected with hepatitis B and C viruses. Bioneer is committed to doing its best to contribute to human health. For more information, please check out other related videos or visit our email and website.